This is a molecule of DNA during DNA replication. During this process, helicase, which is located right here, splits the connections, or the hydrogen bonds between the nitrogenous bases connecting the two DNA strands. The DNA is tethered, which is represented by this clip right here, so that when the two strands are being pulled apart, the DNA becomes supercoiled, as it does not unwind. It just is being split apart. And this is due to the lack of free rotation, or the tethered bit of the DNA. So as helicase continues to split the hydrogen bonds between the nitrogenous bases and move down the molecule, supercoiling occurs. This continues to occur and will not stop until the DNA is cut. If free rotation was allowed, then the DNA would simply uncoil itself and be able to move the rest of this molecule by unwinding itself. Since DNA molecules are so long and so winded, and they don't have free rotation, there's the need for the enzyme topoisomerase. What this enzyme does is cuts one side of the DNA strand, and that allows for the single side to unwind itself from the other side. So now we have our two strands of DNA that are no longer supercoiled. After this process, this portion of the DNA strand is able to reconnect to where it was originally cut, and therefore the torsional stress is gone and there's no longer any supercoiling. This continues down the entirety of the DNA strand as DNA replication occurs, so that the DNA is able to break apart as it's being replicated, so that it does not end up knotted together and not being able to finish replication. The end goal of DNA replication is to have two new molecules, but this would be difficult if, due to torsional stress, these molecules became knotted up and unable to separate from each other. This is why we need topoisomerase and the ability to cut one side of the DNA molecule so that they are able to efficiently separate.